Good afternoon, everyone. My name is Ansh Patel. I am a third-year undergraduate biology major, and through this Catalyst cycle, I have the pleasure to work with Yakendra, as well as Dr. Maynard and Dr. Wynn, on revolutionizing the way preclinical testing is done for monoclonal antibodies. Through our Texas Health Catalyst Bootcamp and the lessons we've learned, myself and our fellow associates have been able to assist the team on identifying what truly differentiates them, as well as exploring different business models that he will then present on today. I'd like to thank everyone for being here today, as well as the Catalyst team. And now I will pass it on to Yakendra to present his innovation.
Thank you for having me here. My name is Yakendra, and today I'll be talking about our antibody engineering platform. Antibody therapeutics have revolutionized the way we treat many diseases, and the number of antibodies that are entering clinical trials is increasing. However, despite significant investment of time, effort, and money, more than 83% of antibodies end up failing to get approval from FDA. And one of the major reasons for such a high failure rate is our reliance on preclinical mouse models, which often do not accurately replicate how an antibody is going to perform in a human system. Because of this, there's a disconnect between what you see in preclinical trials versus what you see when you put the antibody in actual human. This has resulted in such a high failure rate and late-stage failures where you have already spent a substantial amount of money and efforts into developing an antibody. So we have developed, to tackle this problem, we have developed an antibody engineering platform that allows us to engineer antibodies that are capable of engaging with both human and mouse immune cells. This bridges the gap between what you see in preclinical murine models as well as human clinical trials, and also provides an enhanced predictability of efficacy and potential side effects of an antibody. Simply put, if an antibody is going to fail, it's going to fail early and it's going to fail fast. That saves cost and time. The antibodies that are engineered using our system can be tested in readily available mouse models. That not only saves cost, but also time. We have a team of experts in biochemistry and immuno-oncology. And thanks to our mentors, we have been really able to reshape our commercial strategies as well as fine-tune our scientific approaches moving forward. So we have already successfully completed in vivo studies involving human and mouse immune cells and have already secured a provisional patent for this technology. And we are currently working on conducting an in vivo mouse model study in a melanoma model. So moving forward, we are planning on expanding this study, as well as we are seeking strategic partnerships and collaborations with CROs as well as biotech companies. And we're also looking at a possibility of establishing an in-house preclinical antibody testing system. We truly believe that our product has the capacity to revolutionize how antibody development process is right now, and we hope you join us in this journey. Thank you, and I'll take any questions.
Hi, good presentation. So you would be using this in discovery, but it probably wouldn't be in the final therapeutic product?
Yes, that's the thought.
Okay. So how would you prevent anybody from using this technology without licensing it from you?
So we have already filed.
Mm-hmm. Right, but just because you have a patent doesn't mean that people won't just use your idea without telling you that they're doing it. I haven't explored that. Okay, thank you.
Looks like we're good. Thank you.
All right, your market size. So how big is your market for this, that R&D market?
So that's a great question, right? So I've had some conversations with representatives from CROs that are involved in supplying these mouse models. and they're just waiting on some extra, in vivo results, and they're very excited about this. We are going to be working with the Office of Technology Commercialization at UT to discover exactly what the market is. But most of the antibodies that fail, they have gone through the in vivo testing process, right? And we know there's a clear problem. So we think the market is enormous. We just haven't quantified it yet.
Thank you.